So I think I might be online. Let me quickly check. Let me check. Yes, it works. I'm always happy when the technology works. Yeah, hi everyone. Hello and welcome again. Microbehunter here. I'm Oliver, and welcome again to another microscopy live stream. I almost wanted to say microscopy evening, but of course it's not in the evening everywhere around the world because, yeah, as some of you already put down the local time here, it's for example 2:30 a.m. in Vietnam, in 2025 in the evening in England, yeah, so 21 half past 9 in the evening in the Netherlands. So hello all around the world, and I have to say I'm really always very happy to see that some people are really staying up quite late or quite early in the morning to join, right? Yeah, I'm going to just spend the first couple of minutes just to say hello all around the world, simply to give people more time to join the live stream. So I'm Norway, Netherlands, USA, the UK, the USA, Athens, Greece, Romania, the United States, Florida, Sheffield, England, okay, London. So that's really nice. And Netherlands, Carinthia in Austria, in the Philippines. Sound is and video is good. Thank you very much. From Brazil, from the UK, from Slovenia, from Upper Austria. Yeah, hello also from Upper Austria. Okay, so yeah. So what I'm going to do today is the following. I've got a whole bunch of how do you say water samples here, right? With not only just water but also algae, and it's time to do some cleaning up, right? So what I would like to do is I would like to put most of them under the microscope today, simply to have a look, and then I decided not to throw it out like I indicated in the title of this live stream, but actually to put it to collect all of it together in this jar here. So I'm going to simply dump everything together, and then I'm going to see what happens over the next couple of weeks, right? Normally you want to always keep the water samples separate, I understand, because you don't want to cross-contaminate, because from different bodies of water you have got different microorganisms. But I say, who cares? Before I just dump it down the toilet, I just might as well dump it into the same jar, and then let's see what happens over the next couple of weeks. So this is a little bit what I wanted to do today, after I've considered a little bit some other topics. So I'm always very happy if you have any suggestions on what you want me to do. And maybe later on, if you have some time, if you joined in last week, you remember that I've made those microscope slides with diatomaceous earth. We can also quickly put them under the microscope, but I can tell you right now that they do look very similar. Okay, so there is not such a big difference, as I expected to be. So we can do that later. So if you are new to this format, please do feel free to join the chat as well. But if you have any specific questions, then please indicate this with @Microbehunter or @Oliver, so that it's easier to find. Off-topic questions I would like to answer after approximately 1 hour. Okay, so yes, there is a question that I want to address right away, because this has already been asked before in also one of the community posts. Is why is it that some of those are sealed and others are not sealed? Okay, so I'm going to show you a couple of, it's a little bit of an experiment. Normally, when you collect water samples, what you want is you want it to keep it open. That's, I think, really important. Otherwise, there is the real danger that the whole thing becomes anaerobic, especially if you fill it up all the way and put a cap on it. That's a really bad thing to do, right? It's going to, after a few days, 2-3 days, it's going to start to smell, bad, foul. It's often like rotting eggs, like hydrogen sulfide will start to form because it's decomposing, and this is going to kill off a lot of microorganisms. Anaerobic bacteria are going to start growing. So generally, you want to keep it open, and when water evaporates, it will evaporate. You always keep on adding a little bit more water. Now, some of those are closed. And some of those samples are fairly new, and I closed them, and they are still generally okay, right? And the reason is because I made sure that there is a lot of air on here. So it's a little bit of an experiment that I'm doing, right? So generally I would say keep it open, right? So I would say that this one is already a little bit too much water here. Yeah. So this one over here, there is also one sample. I don't know where it is. Where which I kept closed. This one over here, I'm not going to put under the microscope. It's alcohol. It's a leaf here, so I have to keep it, of course, closed because otherwise the alcohol is going to evaporate. So I'm going to put this away because this one is not going to be part of the samples here. Okay, so let's put it over there. Yeah, you can see that there is also some algae growth happening here, and the sample looks now very different than from a couple of days ago. This is actually a pond that I collected, and now you can actually see that some of the not only algae but also cyanobacteria and other started to also grow here. In sheets. So essentially, this one over here I kept closed as well. That's some marine water. This one I collected, I don't know, over a year ago. Yeah, in Croatia. Honestly, it is not something that you want to do, right? It's not going to be. I don't expect much alive here, right? So you see, it's a little bit of a semi-formal experiment that I'm doing. Simply some trial and error stuff. But I have not looked at many of those samples for quite some time now. So I'd like to do that. So that's basically one thing. Before I start a few other things here, I kept 3 jars of water fleas and they died. Yeah. So so that's one of them. So there were a whole bunch of Daphnia in here, and for whatever reason they died. Maybe I didn't feed them enough or overfed them. I don't know. Maybe also because of springtime now, maybe too much sunlight could also be a factor. Over here there were in this one over here there were a whole bunch of ostracods. Yeah, also not present anymore. You can see that actually there is some duckweed growing here on the top. So what I'll be simply doing is I'll be dumping everything in here, and then I'm just going to keep this as a little mini aquarium, and then we're going to see actually what happens. Okay, so that's one of the things. So this is going to be a little bit a trial and error thing, and some of the specimens might not be super interesting. So just be forewarned, right? But that's a little bit also the format of this live stream that I don't want to be unrealistic about the thing. So sometimes I put stuff under the microscope and you don't see anything at all, right? So, and then this thing here, this little contraption here, which I 3D printed today. And it's basically, I quickly designed this, and that is supposed to be a tube holder, 45 degrees. Yeah, it looks like this. So this brown stuff here, that's simply some old 3D printing filament. It's not dirt, just ignore that. And I glued it together with nail polish. In one of in a community post that I published today, I actually show you how the different parts are. And why did I do that? Because if you look at many of those, these are centrifugation tubes. Some of them have a flat bottom and they can stand, still unstable, right? And some of them have a conical bottom here, right? And they cannot stand at all, right? So these tubes are used for centrifugation. So if you want to, I don't know, isolate some solid material, you put it into a centrifuge, and then you have it collecting on the bottom. I like them a lot because they're watertight. And they're practical, and they also are transparent. That's really important because you want light to be able to go in so that algae can grow, and because they make oxygen by photosynthesis so that the microorganisms have enough oxygen. So that's really important. But this is super unpractical. So some time ago I 3D printed those racks here. If you wonder why it's made of so many different colors, this is because I simply changed the filament and while printing. So I simply used some for actually taking out individual samples, I decided to do something like this here, right? So this is how it looks like. And so I can simply go in with a pipette and take some samples out. So this is a little bit the thing. I would say cardboard also works, or some plywood or whatever. I just 3D printed it because I've got a 3D printer. But I would say that this is let's see how well it works. Never used it before. I just made this a short time ago. So every now and then I'm going to quickly pause, and I'm going to read some of the comments. Okay, specifically. I'm going to always jump to the things that say @Microbehunter, okay? So Jesse is saying, I love the simple 3D printed stand from the cover image. Yes, here it is. I've really done this very quickly. Maybe a little bit in the sense over-engineered, because 3D printed, you probably want to use cardboard or some kind of, I don't know, plywood or whatever. Okay, hello from Germany, from the Tennessee, United States, okay? Okay, there's some weird delay on my TV's YouTube. Ah, interesting. Uh-huh. Also, some of you are watching this on television even. From Rome, okay, from Canada. Okay, so this is now, the question is why some of them are sealed, yeah? Um, yeah, and yes, I will try to answer it. I will do the following. Those questions that are on topic, I try to answer as far as I can do that. Yeah. Did you 3D print the racks? Yes, I did. Yeah. So those racks, I actually also 3D printed. This took quite some time. Yeah. So there are also several parts. I don't know. Maybe I should go up here a little bit. Yeah. I show you another commercial rack. Just a second. So this here is a commercial test tube rack. I'll just show it to you like this. It's also made of plastic, and I kind of use this a little bit as a basis, as an inspiration, right? And then I basically 3D printed those here. Those tubes here are a little bit smaller than 3 centimeters. I think 28 millimeters across, and the diameter of those here is 30 millimeters. Yeah. So this is how it looks like. Yeah, so and I also made a little hole here, so for those for the tip here, so that you get the idea. It's highly improvised, the whole thing. So I'm just gonna do the following. Yes. Before I will answer this. Did you get my email with a free PDF link of the very nice guide and key to ciliates in English for that who keep asking? Yes, thank you very much. This is this was a link to a PDF by Mr. Feusner. He was a protozoologist from the University of Salzburg, also in Austria, so not all too far away from where I am. He was a very world famous protozoologist, and he made many of his books available free for download on his website as well. And I don't have the link now with me, but I will put it then later on into the description of this video. And there is a nice, there are several nice books that essentially are quite useful if you want to dive in deeper into protozoology. So I'm just skipping over the off-topic questions for now, if it's okay. About the question about filamentous algae. Yep. Okay. What's the title of it? Unfortunately, it's on my other computer, but it's on the. It's called freshwater biology, I think. Okay. It's freshwater biology. It's called, and by Mr. Feusner, Foiss. N-E-R, Feusner. And if you actually Google him, he's got a separate website. He already passed away a few years ago, unfortunately. But yeah, he was a very active in the field of protozoology. So that is, so Mr. Feusner was from the University of Salzburg. He was quite active there. I will study biology in Salzburg. Yeah, that's a great place to study. So, you know what? I'm talking too much. I just realized, and I now need to find my slides, which I kind of cleaned today and washed today, and now here they are. Okay. So I hope I got them reasonably clean. Okay, and we're just I don't know how we're going to do that. I'm going to just put those here away for later, so that I clear a little bit the space. And we're just going to start with this one. Okay, so this one over here contains sand. Okay, this is some. You see, I made a mistake. Okay, the mistake is the following. Look at this tube here. What do you notice? Big mistake. You, as a scientist, or you never. It's not labeled. How in the world? Yeah. Because I said this, who cares? I'll just basically dump it away anyway after a short time. And I did not throw it away. Yeah, I just made a couple of videos. I documented all of that. And I just kept the sample. I forgot from where I have it. Right? This is, always label your stuff. Honestly, seriously. Yeah. So next thing, what I'm going to do is there are sand grains in here. Sand grains are too large. I'm not able to pick it up with this pipette. I'm going to be using this pipette here. It's a 50 microliter. You can adjust the volume over here. Yeah, simply for convenience. I have a whole bunch of disposable pipettes as well. Yeah, who cares? I'll just be using this one over here. What do? You probably already know. If the tip is too small, you take a scissor, yeah, and you simply cut it off. You make sure that it's not squeezed together, and then you try your best, yeah, and you go in here and you take up a sample, put it on a slide, and what I normally do first is a quick assessment. I will not put on a cover glass. You got to be careful a little bit because that you don't get your objective wet. But I'll be only using the low power right now, so it'll be fine. And yeah, if there's nothing interesting on here, then I simply am going to dump it out, right? So what I have to do is I have to go to the scope view. Here we are again. Here we go. Yeah, for those of you who were basically part of the live stream a couple of weeks ago, I was really stressed because all of a sudden everything was interrupted because I had some USB problems. I think not anymore. I'm really happy about that. So this is how it looks like. Yeah, and usually what I recommend is that you always look for some movement, and there is some movement going on. Yeah, so this sample is already a couple of months old. I'm serious. Yeah, and here we go. Yeah. So there is still it's still alive. Yeah, so there are tiny whatever this is. Yeah. Sand looks quite nice. In case you wonder why the blue background, are these some kind of filters? No, this is a special technique called DIC. Also something moving over here. You can change the color around. I'm just saying, some people want to know. It's kind of an expensive technique. You can also use color filters, but how is it going to look like with under quotation marks normal? That's basically, these are now the natural colors. Yeah. And if you, so the question is what I'm going to do with that. Yeah, keeping this is not really worth it. On the other hand, I don't know. Should I just dump the sand into this? You know, that's what I said. That's what I said. What I'm going to do. I'm just going to dump everything together, right, including the sand and everything, and we're going to see what happens. Yeah, this is definitely going to be a shock for many of the microorganisms in there because it's a different type of water. Yeah, who cares? We're just going to do the following. So we see that there is a little bit of life still present. Okay, but certainly not a lot. Sometimes the movement is so slow that you need to observe it on the time lapse. Yeah. So this is first sample here. Yeah. Occasionally I'm reading the comments here. Yeah, some of you want to know about culturing of E. coli. Yes, I can do that as well. Only E. coli, seriously, because there are some legal guidelines as well. I don't want to, and honestly about culturing of bacteria, I've mixed feelings because I'm kind of worried that other people are going to copy that, and then they end up culturing stuff that you shouldn't be culturing. So this is a little bit my concern, right? Yeah, while plants green, I can talk a lot about this. This is actually an interesting topic. Yeah. So look what I'm doing. Look what I'm doing. No, I'm not going to be doing that yet. No, I won't dump it in yet because I first have to put this under the microscope, right? I want to see what's in here. Huh? So let's do that first. I'm going to take a large pipette. Let's stir up some of the, so it's green. I stirred it up and let's see. This one over here. I am going to put now a cover glass on top. So why are plants green? It's that's an interesting question. Essentially, the sun's solar spectrum apparently does contain quite a bit of green color. So from an evolutionary standpoint, it would have made sense if, because plants mostly use red and blue light for photosynthesis and not so much green. And however, there doesn't even seem to be that little green color in the sun. So I don't know what the exact reasons are. And indeed, I have also read somewhere that scientists are still trying to figure this out a little bit. Because apparently from an evolutionary standpoint, because apparently the solar spectrum does contain quite a bit of green color. So it would make sense that plants also use green color more for photosynthesis. Yeah? Plants are green because the green color is rejected by the plants. But then again, it works. And that is essentially all that nature cares about. Are the organisms able to survive? So this seems to be a fairly uninteresting. Look at this. Those, you find them all the time, those fibers, microplastics. Yeah, it's all over. You pull a water sample out from somewhere and you find those fibers. Of course, it could have been already on the microscope slide. You always have to be a little careful about that, jumping to early conclusions. But even if it's really clean, in a water sample, you always find those things. Yeah, very often these are some kind of synthetic fibers from clothing, for example. Yeah, so they found it in air, not only in air, in rain. Yeah, so you see, disappointing sample. Well, what do you mean disappointing? I mean, some algae, whatever, but I was kind of hoping for more, interesting stuff. So I think, we now know that in this jar there is not, at least from the sample that I pulled, there's nothing super interesting in here, right? So what I'm gonna do is the following, and then if I keep on being that slow, then I will not be able to put over 20 of these tubes under the microscope. So look, I'm going to do something completely outrageous. So this is fresh water, okay? So I'm not going to be mixing salt water and so that's important. Yeah. So look what I'm doing. Outrage. Okay. Yeah. The rest goes away. Trash can. Fine. I could actually also rinse it out a little bit, but then I'm gonna get myself a brush maybe, and I'm gonna rinse it out. So this is the first one, and this one here goes because people also wanted to know where does all of this stuff go. Okay, I'm gonna show you. Where do these go? Those slides, they go into my plastic box to be washed. So what I do is I pour hot boiling water over it to kill everything off, and then I kind of wash it. Yeah. You can also, if it's really difficult to clean, you can also add some how do you call this? Some dishwashing detergent. This sample here I just collected quite recently. There is a small rock in here because there was some algae growing on the rock, which I had problems getting off, so I just took the whole rock. So let's have a look at this. Okay. Now I am going to change the tip around, and let's see what we have here. Okay. So let's go in here. Do this very aggressively and just give it a little try. Okay. And so, by the way, I don't know if you're already into microscopy or not, if you're just watching this, observing water samples is actually a very common thing to do because there's often very interesting stuff in there. But again, if you're using those water samples that I've got here, sometimes standing on my window now for I don't know how many weeks or months, then you should not be surprised that the biodiversity is not specifically very high. Reason being that there is usually a progression. Of organisms. So what we have here is, I've seen some diatoms. Yeah, these things here, these are of course diatoms, those tiny things here. Yeah, okay. So this of course begs the question, should I not try to extract those diatoms as well? Look, there is some kind of something that seems to be moving here. It's also one of the reasons why I'm also showing you those samples is simply so that we all, I myself getting included, get an impression how different they can be. Sometimes there are quite a lot of diatoms present, sometimes more filamentous algae, sometimes more cyanobacteria. So each one of these tubes is a little bit like a separate ecosystem. So I mean, I'm now at the 20 times. You see it up there. That's the second objective. The third objective is the 20 times objective. So what I'm going to do is occasionally I'm going to read again. Okay, I'm just going to skip over many of the questions if they're not directed to me. Hello from the United States, from North Carolina again. Okay. Yeah, I will Google and check. There's still a debate, yeah, about the colors used for photosynthesis. Yeah. So, yep. Okay. Yeah. So this is just some discussions going on. Yeah, so concerning photosynthesis, they're green because chlorophyll is green, obviously. And now there was, of course, I used to think that, well, it's because plants are evolutionarily adapted to the solar spectrum. But then I read somewhere that actually sunlight contains quite a bit of green light as well. So it would kind of make sense if plants also were to use green light more. So the question is, why don't they do that, right? And as a matter of fact, they will use a little bit of the green light because there are so-called accessory photosynthetic pigments like carotenoids, which are orange, xanthophylls with a yellow. Then you've got chlorophyll A and B. Okay, there are two versions of green. There's also pheophytine, which is slightly greenish, bluish. So there are a whole bunch of pigments besides the green chlorophyll as well. Look, that is a moving diatom. Here we go. Okay. Yeah. So they have a gliding motion. Yeah. So they're moving along in a gliding manner. Yeah. Now at this magnification, we also see some other tiny microorganisms. Okay. Two diatoms. Nice. Yeah. Yeah. Let's dump it. Let's dump it into the jar again, right? As it is. Yeah. And let's go back to the desk view. And again, something you shouldn't do. Let's cross-contaminate. Blup. Yeah, and this one, I think I'm just going to keep this tube here. And I'm just going to wash it out and then take a brush to clean it. Let's put it over there. Next one, what is this? This looks, yeah, this seems to be also some kind of a biofilm now forming here on the side. Yeah, I guess I'm just going to use the same. I'm just going to use the same pipette. Who cares? There won't be so many differences. Won't be too much. So yeah, now I can use this again. Now you actually see it actually in action. So lots of green stuff here. Now, this seems to be quite dense. So sometimes it's a good idea to kind of break it apart a little bit. And yeah, I'm also, by the way, because I'm doing so much microscopy, I'm also recycling the cover glasses. Yeah, so I also wash them. And by the way, I've got now an ultrasound cleaner. So I've tried it also already, but I'm not so sure of whether it's worth it. Okay, so I still will do an experiment by how do you call this? Heat fixing some bacteria or some yeast cells, maybe some fungi, and then see if the ultrasound cleaner is able to get it off. A scope. Here we go. That's the dense one. Okay, here we go. This is a little too dense. Oh, this is a little too bright. Let's close this. Let's turn on the DIC again just to get a little bit of color. See, this is the problem when the sample is too thick. Yeah, then you have problems focusing. Okay, here are lots of diatoms, right? Okay, this seems to be slightly more active. You see there's more movement going on here. Yep. Whoop, there's some this one is a little more active. So, some months ago, what I have done is, I think also in one of I already forgot what I've covered in my live streams. I've extracted those diatom shells, the frustules, by mixing the sample with hydrogen peroxide, and this digests everything away that's organic. And then you've got those nice diatom shells, walls left over. Actually, you shouldn't be calling them shells. The reason is because shells are made of calcium carbonate. These are actually silica. So basically, they're basically called frustules, and they are basically made of silica, so of glass. And when you mix the whole thing with hydrogen peroxide, then all of the organic material is digested away, and then you have those nice, under quotation mark, shells. The cell walls, in other words, left over, and they look kind of nice. So you see, this one again is different. Yeah, I mean, I'm not going to do that now because I've done this so often before. Do you see those bubbles? Yeah, those bubbles are going to grow in size because these are, of course, oxygen bubbles that are formed by photosynthesis. Yeah? So I'll be dumping this one as well, but I will read again. Yeah. Yeah. That's an interesting. Chlorophyll might have evolved underwater. How deep does green light penetrate water? As a matter of fact, the reason is it's indeed the case that for example, red light is filtered away and from the water, and this is the reason why, for example, plants that are deep water plants that are deeper down, certain algae have photosynthetic pigments that are more red in color. Indeed, because the water plants deeper down, they will receive a different light spectrum, and that means they are more brownish in color. So that's the brown. So that's why some algae are brownish, simply so that they're able to absorb more of this of the greenish light that is able, or bluish light that is able to reach the bottom of the ocean floor. So that is actually correct. That's one of the reasons why algae further down or water plants that are further down in the ocean are sometimes often more reddish in color than greenish, because they receive a different light spectrum. Yeah. Don't forget to label the jar. Yeah, exactly. At least I'll put a date on it so that I am able to monitor the progression here, right? What micropipette capacity? Okay, that's an off-topic question, but I have 2 pipettes. Okay, maybe I should talk about it later, but who cares? As I go on, look, this one is a milliliter. Yeah, so these are they take. I don't even have them here right now. Oh, here they are. Okay, here they are. So these are the big tips for the milliliter, right? And I rarely use it because it's way too much. Yeah. It's so what I find much more convenient are those. This one goes to 5 to 50. Yeah, I think because that's pretty much the same amount that in the range that you put on a microscope slide. So I found this to be very practical, and it also allows me, if you have a very steady hand, it also allows me to actually pick up individual, let's say, tardigrades. From the microscope slide. You do have to have a very, very steady hand. And something I also tried once is by heating up the tip over a flame, you're able to pull it out, and then you're getting a very thin tip. Okay? So I just wanted to, I just did that. So look what I'm doing now. Again, and bloop. I'm cross-contaminating. Look at the biofilm in here. Right, lots of interesting stuff still. Yeah, I need to do this cleanup because otherwise, look at this here. No go. This one has already dried up. Okay, I forgot to add some water here. So this is you can see how. Yeah, nah, I'm just not gonna use this here. Okay. You know what? Why not? Why not? Who cares? I'm going to. I don't have my Petri dishes here. Look what I'm doing. Let's do the following. I don't even remember where I have that sample from. And you know what? I do have a stereo microscope. Does it work? Does it work? Yes, it works. Yeah, here are 2 snails. Yeah. Just for the fun of it. And here are 2. And here, this is the sand sample. It's still moist, so maybe some of them have survived. I don't know. Nah, it doesn't look very moist. Yeah. So but obviously that's how it looks like, just because I happen to have it. Yeah. You might find crystals in the dry material. That could be. Yeah. Yeah. So, but I'm not going to be putting that otherwise under the microscope. Let me simply throw it away here, and okay. And the next one, what is this here? Oh, this one is empty. I don't know what this is. This is empty. I'll wash this out here. So these are, I think, some slightly more, I don't know if this is some slightly more recent samples. Yeah, here I've got also a rock in here. And let's see if there's something in here. And then, Sometimes always make sure that you take stuff from the bottom because yeah, it sometimes settles down, and then you want to agitate it a little bit, and you want to take some solid material as far as possible. And so we throw this away. So let's have a look again. Scope. Ah, okay. Also some gliding diatoms. Oh, what is these? These are some dead water crustaceans. Looks like. Let's go down with the magnification a little bit. There's something is moving here as well. Okay. Yeah, these are basically what you see over here, these things here. Yeah, these are some dead. It could be some kind of an ostracod, maybe, I don't know. And that's also something that I've already seen before in those dead animals that you have here. Sometimes you see on the inside, you see some ciliates or flagellates, some cells, and so I think they're just living in there, eating it up or something, right? So yeah, this is something that I've already seen happen a few times. You see somewhere a dead animal, and then inside this dead animal, the outside, the exoskeleton is hard, right? And then there's a couple of cells in there. Either one of them found its way in there and started to divide in there, but I think they're just eating it up, kind of, from the inside. Yeah. So this is actually quite. Let me go up with the magnification. It's further. Here we go. Let's turn on the. Yeah. It's actually quite a. Yeah. Just recently made another video about this, yeah. So that's basically a couple of cells caught, a couple of cells caught inside, a dead water crustacean. Yeah. I'm just reading again some of the comments. Are you gonna throw away the jar's contents? No, not yet. The jar I'm just going to keep. And I'm going to allow some kind of a natural development happening. Some microorganisms are going to reproduce more, others are going to die off. So I'm just going to see what's going to happen. So what I do is I simply, it's a little bit of a highly unorganized experiment of seeing how what actually happens to the jar. Because the water fleas that I had in here, the ostracods and the daphnias and so on, they all died. Maybe I didn't feed them enough. So I just said this before dumping it out, I just might try a little experiment. So this one here is also quite interesting in that sense. Yeah. So what I'm going to do is I'm also going to dump this one here. Where is the desk view again? I'm really happy that the computer system with the cameras now works kind of reasonably well again. So I'm just going to agitate the whole thing, and then 3, 2, 1, blop. Yeah, here we go. So this one will also be washed. Next one, that's one of those. This one I collected last week. Okay, from the river Ybbs in Lower Austria. For those of you who know a little bit about the geography of the region, it's and here also some algae, and let me see what I'm able to find here. Now, this seems to be a fairly dense sample as well. So let's have a look here. This slide goes out. I always start with it. You're not seeing this. I have to switch over to the microscope view first. Here we go. Oh! Clostridium it's called. Not Clostridium, these are dangerous bacteria, but Clostridium is actually a nice algae. Clostridium algae. How can I make this a little bit brighter? And this is actually a very nice. Nice one. Let's go up 60 times. Let's adjust everything here. Okay, here we go. That's a nice find. Okay. Oh, seems to be another one. And one of the typical things I just also want to tell you that are those little, and I think they're also not quite sure. Scientists also don't quite know what the purpose is. But do you see those at the edges here? Those moving, I think particles here. That's also very typical of them. But I don't know why they are there, what the function is. Yeah. But those tiny little dots that you see here, on the side. Yeah. But this is such a nice specimen, and I'm right now at 60 times. I'm not 60 times. And I'm going to be dumping this one into the jar. What a pity. You know what? I think I'm just going to try something that I normally don't like doing. I'm going to go oil immersion. Heavens, why am I doing this? Okay, so this is I'm going to go now the maximum magnification, okay? Because it's simply such a nice specimen, and I just would like to, before I dump it, have a nice video of this. So this is, you gotta watch what I'm doing here at the top now. This is now high risk, okay? Because I really have to be careful that I'm not damaging my other objectives. So number one, I'm going to now, and you cannot see that. Let me see this. No, you can't see that. I have to now set the focus lock lever on the microscope. So the focus lock lever has been set. I just am going to demonstrate what this does, okay? Look what I'm doing. I'm now, look at the top. I'm lowering the stage, right? It goes out of focus using the coarse focus knob, and now I'm raising it again. And plop, it stops here. Yeah, it's locked into place, right? So it will remember the position, right? So, and that is, one of the nice features of the thing here. And what I'm going to do now is I'm going to, look what I'm doing right. I'm going to apply now a drop of immersion oil. This objective over here, that's the oil immersion objective. Only this one is allowed to get in contact with the immersion oil, right? Yeah, only this one. So I have to apply, no, I'm going to lower it first. Okay, lower it first. I put it into place. Drop of immersion oil goes on top. Ooh, I touched it. Dang. So I moved it. I now lost the how do you say, the algae again, because unfortunately I touched I moved the cover glass. So I'm going to now go up again until to the maximum level. Okay. I need to move this back and forth a little bit like this. Unfortunately, I moved it. This is a pity. Yeah. Okay, this is a pity. Okay, so I'm right now at a how do you say, at 100 times oil immersion. Yeah. See, that's one of the risks that you have. Yeah. Because yeah. Yeah, there is also a little bit too much water present. See how difficult it is to actually find. And as I'm focusing back and forth, do you see the movement now? Yeah, that is I'm focusing, and the oil applies some slight pressure on the cover glass, and this causes the whole thing to move back and forth. So this is a little bit of a pity, but it kind of shows also a little bit some of the, and you see how high the magnification actually is. Totally blurry. Yeah. Did I lose the oil? Yeah. Looks like. Yeah, so when you do oil immersion, you have to really make sure that there is only very little water. Yeah. You know what? I'm gonna give it another try. So let's start from the beginning. Okay, we lower this here. Let's remove the slide. Let's take the desk view again, and we're gonna start from scratch because I wanna get a nice picture of this again. So, and now with very little water, very few algae. This, I don't know. I want to see some green stuff here as well. So this is still a little much, but if there is too much water, then if there is too much water, there should only be one cover glass, then yeah, if there is too much water, then you use some tissue paper to remove the excess. You do not want to have a swimming cover glass. It's a little bit the cheap way of doing it. And you just got to make sure as well that there is not too much water on the surface itself. You don't want to mix it with the water. So this one is actually very thin. And now the cover glass is also fairly solid. So I have to go again to the low magnification, the square, the scope view here. So let's give it another try. Maybe we're lucky in finding, I have to find something of interest first. Yeah. Oh, this is, now I got too little sample. That's a pity. Let me quickly check here, directly under the microscope. Maybe we'll have a look here at this here. Okay, this is now at 20 times, 40 times. Okay, let's have a look at, I think Patsillaria is the name of these diatoms, here. So 60 times. Okay, and let's have a look now. Let's go oil immersion. Okay, let's have a look at this under oil so that you also see how this actually looks like. So I'm focus lock lever, I'm lowering this here, I apply a drop of oil. You have to change, never forget to change the objective, that's really important. So, and here we are. This is now maximum magnification. Okay, the depth of field is quite low, but at this magnification at least we're also able to see the nice structure of the diatom shells. I love it. A little fellow over here. Using gray background DIC, I can change the color here as well, of course. And I have also another possibility to adjust. Down here, this is also a polarization direction that I can also adjust here. Let's see. In any case, that's now maximum magnification that we have. Look, it's still moving. When I focus, then it's moving back and forth because of the slight pressure that the oil and the objective exerts on the cover glass. So open it to 100% is requested. So that's basically now opened completely. The correct. And but right now the contrast is a little bit lower. Depth of field is also, of course, much lower. But you get the idea. So this is actually probably, concerning the diatoms, actually probably one of the nicer specimens. Here the cells are all clustered together in this matrix, in this biofilm matrix. Gray background. You want to have gray background. Okay, let me see if I'm able to adjust it to gray. I have to find it first. I don't know. I gotta find it first. There is a setting. Maybe this goes a little bit more into the grayish. So what I can do is it's still what I can do is I can adjust the prism on top, and I can also adjust a setting on the stage, and I can therefore adjust the colors. This is a little bit more a neutral gray. I'd have to find it. But yeah, here in DIC you do, and that's has been a little bit requested, is you do see a slight this three-dimensional effect. Yeah. Yeah. So this is actually, Marcel is asking about immersion oil. Has a refractive index of 151, so the cedar wood oil, for example, but I imagine that it takes more than just a refractive index. Okay, I will tell you the following. Why I'm using this immersion oil. That's the one that I got with the company Olympus. And it is a so-called a synthetic immersion oil. And it is completely clear. It is the cedar wood oil, immersion oil, is yellowish. I don't like to use the cedar wood oil because it's a natural immersion oil. That's not the problem. But the problem is the yellowish immersion oil, the cedar wood oil, tends to solidify over time. So this means you got to wipe your objective, otherwise it's going to turn hard. And I've had some of the old bottles of immersion oil that actually the oil was completely hard. And this does not happen with synthetic immersion oil. So for me, it's not only a question of refractive index, but also a little bit of whether it actually solidifies or not. And another thing that has to be taken into consideration is that sometimes there are certain additives in the immersion oil that are not quite healthy. Yeah, so you should try to minimize getting it on your fingers. Yeah, I'm getting carried away. Actually, I wanted to So the condenser is now all the way open. Okay, so I'm now using the adjustment below the stage and here on top of the stage. You can play around with both. So I'm going to now do the following. I'm going to clean it later. Okay, I'm now going to simply remove this again, and let's move on to the next one. This goes over here, the desk. So the oil goes away. So this is the one here, and blop. Yeah, here we go. Next one. What is this here? Not labeled. Of course not. Of course not. I don't remember where I got that from. What is this even? There seems to be some solid stuff in here as well. So let's take again a sample from the very bottom. And because it's springtime right now, so there are plenty of if I want to get a new water sample, just go out and get a new one. So it's not, that's why I'm so let's just start again over here with low. So where is the scope? Yeah, this is of course way too bright. Not particularly, I don't know. Oh, there's a couple of diatoms. Let's go up here. So. Yeah, you see now the setting is in such a way that I don't have so much the blue. Okay, here's the blue again, right? Yeah, but here it is now the more in the grayish area. Is this? It's kind of small. I don't know. Let me see. What is this here? Oh yeah. So a couple of diatoms again. Diatoms. Yeah. That's how it looks like. So also a few living things in here. I mean, actually a lot, but I mean, yeah. Oh, this one is too fast. Yeah, by the way, I've in case I just recently made a video about fast-moving microbes. So one possibility would be is to add some methyl cellulose. Just recently I made a video. That's this one over here, right? Methyl cellulose is a highly viscous solution. It's actually also used or used to be used for putting wallpaper on the wall. So yeah. So this is highly viscous and has the advantage that it slows down the microorganisms. Oh, spiral-shaped bacteria as well. But it does not have, is not very osmotically active. So let's have I don't know, this might be a little too difficult, but let's try phase contrast. I only have one phase contrast objective, and it's a 40 times phase contrast objective. Okay, then maybe we're able to see the bacteria a little bit better. Yeah. No, because they're also moving so quickly. So this is, yeah. Occasionally these spiral-shaped bacteria, spirilla, which look kind of nice as well. I've seen one before, but not anymore. Oh, this is too quickly. It's not one of those. Yeah, you get the idea. Let me quickly have a look again at some of the questions here. Why our stream starts 1 hour earlier? I'm a little bit confused. What do you mean it starts 1 hour earlier? I'm always starting at 21:30 Central European time. So, yeah. Could you use image stacking to get a larger field of depth of the contents of an algae? The chlorophyll looks nicely packed in a helix. Yes, I have also done this already. It is possible, but you have to make sure that there is absolutely no movement, obviously, right? So what sometimes does work is instead of taking simply several pictures, I just make a video focusing through all the way and then allow the image and then extract the individual frames, simply so that there is not enough time. So in image stacking, you try to combine different focus levels into one image, and I sometimes simply make a video of everything and then extract the images later, right? So it should be possible. I've tried this once as well, but the success really depends on whether everything is really stable. So let's move on. By the way, just for your info, I've got now my dark field filter in here, and the phase contrast filter, and the phase contrast filter works like a dark field filter for the 4 times objective as well. So this is actually now dark field. What do you have here? Yeah. So yeah, let's move on. So this one here is the desk again, and here we go. In this jar, by the way, I used to have Elodia, water plants, and so on. It was a very healthy environment, and then the water plants died. I think the snails might also have played their role, right? Because there were a couple of snails in there as well. So next one here, what is this here? This is from a local pond. Yeah, this is also approximately a week old or so, this sample. And so this is a very dense one. Let's break everything apart here. So, and then I'll start collecting again water samples from scratch, and we're going to restart everything again. So scope view. Ooh, wow. Okay. Just a second. Let's try to. This seems to be pretty active. Yeah. Seems to be pretty active as well. Can I change the color here? Yeah, it's now. Yeah. Yeah. Okay. Yep, you get the idea. Lots of diatoms again. And yeah. Quite an active little sample here. So, and usually, again, I know I said it often, you got to keep on checking because after a couple of days it looks again different. Depends on the light situation, oxygen situation, I don't know, possible presence of certain predators, microscopic predators, maybe. Yeah. Yeah. So far I have not seen a lot of rotifers really. Okay. So, okay. Yeah. But this one seems to be a slightly more interesting sample. Yup. Okay, I'm also gonna dump it. This one is a little slower. I used to also have several fairly large paramecia in some of those containers. Not anymore. Yeah. Okay. Okay, that's how it looks like. Again, what did I say? Do you see this nice blue fiber? Microplastics. I love it. This is a pretty huge one. Yeah. So this is actually, by the way, would be an interesting citizen science project maybe for people who have a microscope to kind of look for microplastics and document this somehow. Yeah. So I've seen also in this sample, what was it, the other one, some heliozoans, which are kind of nice. These are, which are have those spikes, star-shaped, sun-shaped rather, I should say. I wonder if this is also some kind of microplastics. Yeah, okay, you get the idea. Let's dump it again. Dump it into the, where is my desk view here? So, here we go. Let's agitate to make sure that everything, bloop, and in it goes. So what is this here? Ah, yeah, that's the local river where I also collected some algae. Let's do this a little faster now because otherwise this is way too. This seems to be a little more slimy. So put this one here. Yeah. So, and next one. I've got this idea. I wonder what you think about it. I have no idea if it's even a good idea or not. But it would be kind of also nice. It would be kind of nice to I don't know, to have some kind of a platform where you can trade microscope slides. Yeah. But I wonder how this would work. I know there's this book trading platform where you can trade books. Would be kind of nice to also have something like for microscope slides. This one is also, it contains now some kind of interesting creatures here. Let's go up with the magnification again. Ah, yeah, here we go. Here, that's a paramecium or related. Yeah, not the largest one that I've seen. A couple of days ago, I had some really huge ones as well. Yeah. Yep. They seem to be reproducing. Don't do it. I already have like 1,000 slides. Yes. Yeah. I know that would be actually quite nice if somebody, because different people are specialized in different types of specimens. I don't know, maybe some people are more into histology and then making, I don't know, tissue cross sections, and then other people probably more want to make samples of, I don't know, plant cross sections or so. And it would be kind of nice to have them some kind of a platform to exchange them. Yeah, and or maybe from the local environment a little bit. Yeah, what used to be maybe stamp collecting back in the day, maybe nowadays slide collecting. I don't know. Yeah. But this is again, that's again all involved in cost because then you have to mail it around, and it's all very, how do you say, yeah, more complicated. Yeah. By the way, I just want to say something else that I've been thinking about now for, would you just want to share with you again look here, that's nicer, there are more here, for some time. Some of you might actually know that I do collect emails for a newsletter, right? So on my webpage, there is this possibility to sign up for a newsletter, but I have to admit that I have not sent out a lot of them yet. But I do have over 1,000 newsletter subscribers right now. And I've been thinking, yeah, maybe, but the question is what am I going to include in this newsletter? And I was thinking maybe some kind of like follow-up comments or follow-up information, or maybe some recipes, things that I've kind of found out during a live stream. A couple of weeks ago, I did this Beerman funnel, for example, for collecting tardigrades and nematodes. Maybe have some kind of a description on how to do that, so in a more text-based form. So I'm kind of thinking about this. But if you have any suggestions as well, then yeah, please, share them with me. But I have to admit, I've been very how do you say this? Negligent, to get the pronunciation right, about this. So I only have sent newsletters very rarely, and sometimes also with link collections on basically on the videos that are published. But actually there might be the need for, I don't know, maybe some kind of a platform to kind of share also more text-based material. I'm just throwing the idea into the community, okay? So there is a slide exchange called the PMS, Postal Microscopical Society. Really? I didn't know about that. It started in 1873. There is a slide exchange. Isn't this nice? Thank you for the recommendation. I'm going to have a look into that. Okay. Let's see. But this one seems to be quite a nice, it's almost, I don't know. I feel a little bit, it's almost a pity if I were to, you know what I'm going to do, folks? I'm not going to dump this. Look what I'm going to do. There are paramecia in here. It would be a pity. So look what I'm doing. I'm not going to dump it in. I'm going to reach over. Look what I have here. Breakfast cereals, oats. Actually for my slime mold. And one oat goes in here, and that's it. So what I'm doing right now is I'm feeding them, and what these oats are going to do is they're going to grow bacteria, and the bacteria are going to be the food now for the paramecia. So I hope to be able to get more paramecia this way. So this one I'm going to simply keep, right? And I'm not going to dump it together to the others because maybe I'm able to reproduce some of the paramecia here, okay? So next, I still have 1, 2, 3, 4, 5, 6, 7, 8, 9 slides. I've got 9 slides. Let's see if this one here, some time ago I put this one under the microscope, and there were tons of cyanobacteria in here. Okay, so let me try this again. Okay, so let's see. I don't know. Let's see. It's a little. It's I don't know, a little bit maybe it's a little bit. It's not very dense. The specimen sample. It's not very dense. This sample. Oh, thank you very much. Okay, I received again. I again received your questions. Many of which are probably already answered. It doesn't matter. And you know what? I might do. I still have a few left. Otherwise, we're just gonna have a look at the rest next week. Because it's always a nice thing to have something left over for next week as well. Okay. And the next week we can see how far the other thing has developed. So put this away. And where is the scope view? I probably didn't. This sample was not very dense. No, this is not a good one. probably. Do you see those little things here? These are there's some kind of I don't know. It almost looks like crystal growth on the microscope slide itself. These little things that you see here, that's not part of the specimen. Yeah, you have to actually get the air bubbles in focus, and then you know actually where you're supposed to be. But this one is not a good one. Very quickly, I go into phase simply to check if there are bacteria present. Let's see if we're able to find some bacteria here. No, that's basically, I don't know. That's not the thing that I wanted to look at. This is not a good sample specimen. But these could be bacteria. Yep, these are bacteria. Yeah. This kind of also shows a little bit how difficult it can be to sometimes see some of the bacteria. But I don't like this. I don't like this one specifically. Let me quickly try doing this again. Maybe I need to collect some slightly more solid material. Let's go in with a tweezer. Look, we do the following. This is a little bit the how do you say? Yeah, it's a little bit the highly improvised way. I'm doing this a little bit fast, obviously, because normally when you do this, you want to probably take your time. This is highly improvised. So you also want to make sure that it's dry on the bottom, otherwise it's going to stick to the microscope stage. So, and where is the scope? Okay. Let me see. Okay. That's also, yeah, there is some stuff going on here, but it could be more interesting. I think those long structures could be the cyanobacteria. Ah, yeah, of course, yes. Sometimes they're actually moving. Possibly oscillatoria if they're moving or gliding. Let's go up. Ah, okay. What happened here? Okay. Yeah. Mostly suspended stuff. There's some bacteria here. Not particularly interesting. You know what? Let's give it another try by actually taking some of the solid material here, and then I'm just going to leave it, and I'm going to then simply go back to some of the questions, and for the rest is for next week. I'm going to scratch off some of the solid part here. Look. Okay. Let's see. So, what time is it? I always have to check the time a little bit. Yeah, and I'll quickly, after this here, I'll go to the questions and I'll have a look at the chat a little bit. So, here we go again. So, where is the scope viewer? Switching back and forth between scenes is also a thing. Oh yeah, that's way too... It's way too thick. Okay. Yeah, there's a little bubble here. That's way too thick. Okay, here it's better. Okay, yeah, those things here are most likely cyanobacteria, also known as blue-green algae. I know that because I remember it from that tube. Yeah. Not so many diatoms. What is this here? Looks almost like. Sometimes it's when everything's a little more stable, you can take a time lapse, and then sometimes you're actually able to see them move around as well. Yeah, so they do move, and some of them at least do move and glide. There's some little bit movement here. Yeah. Use oil objective 100 times. That's the best, yes, but I do not have a phase contrast oil immersion objective. Only bright field and DIC. But we can still, as a last one, we can still use try oil immersion, because why not? Okay, so let's try. Let's see. I would like to move a little bit further into the center here. But it's kind of dense. Everything's a little too dense here. Yeah, let's have a look at a closer look at a few of those here. Okay, make it brighter. Yeah, these are clearly cyanobacteria. Cyanobacteria don't look like bacteria. Haha, yes. but they are. Okay. See, why is it moving like this now? Because the sample is too thick, and now I'm touching the cover glass. Yeah. So this is actually a problem a little bit. It's a little too thick. Yeah. So I don't know if I have to. If I want to go oil immersion here, I have to take much less. Water. So let's give it as a last try. Let's take a small sample here again. Didn't catch anything here. So a small amount. And let's scratch off something from the surface here. Very little. Let's break it apart. I think that's going to be enough. Cover glass goes on top. Up. And yes, this is now definitely going to work. You see it's, yeah. So the layer is very thin as well. And scope view again. Here we go. Yeah, it's still very dense here, but okay, that might be better. Yeah. See, the liquid is still kind of spreading beneath the cover glass until everything is yeah. So, yeah. Now the objective is not touching the slide. That's good. Open it up a little bit here. So I'm going to now again engage the focus lock. Let's move it down. Yeah, and let's go again, just for the fun of it, to oil immersion. Okay. And we raise it again. Here we go. Yep. I need to change the filter setting here. Oh yeah. And this is also where we start to see some Brownian motion as well. The random motion of the particles here. And yeah, lots of cellular debris, bacteria as well. And these larger fragments seem to be cyanobacteria. Again, it's very bluish. Maybe I can get a slightly more neutral color. Yeah, there's definitely a lot of bacteria here as well. Okay. Here they are. Yeah, always fascinating how different those water samples look like. Yeah. Is it better to use 100 times oil objective or 100 times dry with a long working distance? the question is, what do you mean with better, right? Those two different objectives exist with for different purposes. Generally, if you want to get the resolution, you have to use an oil immersion because it has to do something with a numerical aperture. So there is a physical limit. You cannot get the same resolution, yeah, when you have a large working distance and then also no immersion oil, right? So, but sometimes that is maybe what you want to have the magnification, but maybe sometimes, but you don't need the resolution. So you see, it depends really quite a bit on what you want, right? And I remember when I had a choice of objectives, I bought myself a 60 times objective and then was asked, do you want to have a 60 times dry objective or do you want to have a 60 times oil immersion objective? So yes, there are also companies, some of them offer also 60 times oil immersion objectives. So that the people want to have a high resolution but at a lower magnification. So it really depends a lot. So I wouldn't be able to answer that question. Yeah. Yeah. So, I'm just reading here this again. Okay, so what I'm gonna do is I'm just quickly going to do the following again. I'm going to dump this into the. I'm going to dump this also into the container now. Yeah, and we're gonna see what happens. Ah! Oh, great. Okay, I dropped in the whole thing. Okay, doesn't matter. So here we go. Put this away. Yeah, and now I have this nice, more or less, mix of, I don't know, yeah, of microorganisms that should not be together because they're from different places. I don't know. Oh, did you just see that? There are some water fleas in here. Oh, thank you very much for the donation. Thank you very much. Okay, there must have been one sample in there with a couple of water fleas. Yeah, maybe some copepods or yeah, you see this? There's some stuff swimming around here. So let's see if they're able now to reproduce a little bit. Yeah, definitely. Yeah. And maybe I'm able to restart yeah, a culture here. I don't know. Yeah. Yeah, by the way, last one. You remember this one? This one was the one that I found in this old plastic bottle. Let's do this just quickly as well, so that I can clean out the containers. Yeah. So this very quickly. Thank you again for the donation. By the way, I use those donations also to buy microscope slides, which I occasionally show you. I did buy recently a box of microscope slides of human tissue. But why did I not show them to you yet? Well, the simple reason is because, you're going to laugh, I know too little about it. Because it shows pathological tissues like tumors and all these not so nice things, right? And I wanted to actually talk a little bit about those, but I don't have enough histological training or background, except for one slide where I'm able to explain to you what we were able to see. And for that reason, I have to say it's a pity that sometimes the slides that they have or that they sell are pretty good. Oh, you didn't see this, what I was doing right now. Were pretty good, but there is too little documentation. And that's something what I consider a little bit of a pity, right? So this is now this water sample that from the, where's this scope view? Yeah. Ah, yeah, these were I remember now. These were those tiny algae. Oh, cool. Oh, this was not present before. What are these? Oh. Oh, are these chlamydomonas? Look at the flagella. That would be nice. If this is chlamydomonas, it would be nice. The chlamydomonas, could be related to them at least. Yeah, they have these. or some flagellates with endosymbionts. I don't know. Do you see those? In any case, that's actually I'm quite happy that I still looked at that because this was not in the sample before, right? At least I didn't see them. So look, if you look very carefully, and you see those fine flagella, the long extensions here, right? Yeah, and yeah, so these are then flagellates, or I don't know. Chlamydomonas does look different, but maybe it's a related one. It's green on the inside, right? So I don't know. Yeah. So this is yeah. I know that Chlamydomonas has basically is an algae, a single-celled algae that has those flagella also, and it moves towards light. But one always has to be a little bit careful with simply quickly identifying things under the microscope. Yeah. Yeah. A Winogradsky column. Yes, I've actually also thought about that. A Winogradsky column in maybe one of those tubes here. Yeah, so what you do is in a Winogradsky column, you're growing different types of bacteria. You put in all sorts of paper and wood and whatever, and then you simply allow the bacteria to grow over the course of a couple of months. And then you have in the different regions, you have got different microorganisms growing. Actually, I'm quite happy about this one over here. Didn't expect that. Okay. Yeah, let's. But you know what I'm gonna do now is I'm still gonna dump it together. We're gonna see what's gonna happen because I need to clean up everything, and I'm going to now have a look at a couple of your questions. Okay, at least everything is more or less documented in a video. Okay, so and bloop, here we go. I wonder if some of those are not able or are able to survive the change in water. So, okay. So what I'm going to do now is I'm going to clean up a little bit, and I'm going to have a look at a few of your questions. I think most of them I already talked about or addressed. It doesn't matter. So I'm going to go through this very quickly because actually I spent more time on this now than I planned, but it's fine. So, yeah, some of them are simply comments and not questions here, right? Last week, the biofilm I looked under the microscope wasn't really alive. Yeah. So why were some of the bottles sealed but not and others open? Simply for yeah, no specific reason. Simply, sometimes I simply kept them closed because I wanted to check how long you can keep them closed and then still for organisms to be in there. Yeah. I 3D printed those racks. That is correct. Okay. Yes, I got the email. Did you get your email with a free PDF link? Thank you very much. I will post it then into the comments. Yeah, it's basically a PDF by Mr. Feusner, who was an important protozoologist. Yeah, I would love a video or a stream showing the differences between filamentous algae versus cyanobacteria and how to identify and differentiate. That is actually not, in many cases, not too difficult because filamentous algae are eukaryotes and you can actually see the chloroplasts normally quite well, and they're also much larger. So that is indeed something that I can talk about. Yeah. Yeah, they have a nuclei often visible. Yes, not only nuclei, but the chloroplasts are actually quite well visible in many cases as well. Yeah? And they're generally much larger. Yeah. Yeah. So you think that it's over-engineered? I have to show you my 3D printed one. Yeah, this was a little bit a comment of I think this one over here. Can you please make another video or stream about bacteria culturing in Petri dishes? Yes, and I have yes, maybe, I don't know. You see, I have mixed feelings about this, right? It is. I mean, I'm a trained microbiologist, so I know how to do that. I also know how to actually cook media using household equipment and so on. That is not so difficult, but I'm kind of a little bit worried about the fact that actually I've seen online in YouTube some folks who are kind of playing around a little bit with that, and then they're making fingerprints and growing bacteria and the colonies. and you should not be growing bacteria from your skin. Yeah, you shouldn't be doing that. There's Staphylococci maybe on it. I'm not saying that they're all dangerous, but some of them could be, right? And you're just growing them at such crazily high concentrations. So you see, I'm not so much worried about those water samples here, right? Because yeah, these are yeah, from a pond water and so on. But honestly, growing bacteria that are possibly also have the possibility to grow in humans. I don't know so much if that is a good idea, right? I'm not automatically saying it's all dangerous, but is it really necessary? Yeah, so if at all, then with E. coli bacteria or, I don't know, growing yogurt bacteria, because it's biotechnology, so I have no problems with that, obviously. Free books in German language? No, many of them are actually in English. Okay? Oh, you want to know if there are free books in the German language? I don't know about that. I've got the reason is, look, somewhere over there, I've got a whole pile of over 10 books, microscopy books, all in German. I was actually thinking about doing maybe a special video just in German, just for the German-speaking community, where I just show you the books. But I don't know so many. I've not been searching either so much for free German books, I have to tell you. So I cannot really say so much about that. Yeah? Do you look at any non-biological motifs under the microscope? Crystals, yes, I love looking at crystals. Yeah, vitamin C and other crystals. Yeah, micrometeorites, crystals, and also microprocessors. Micrometeorites I also tried to look at. You can collect them using magnets. However, apparently, just because it looks like a micrometeorite spherical does not automatically mean that it is one. Apparently, the identification of whether it's really a micrometeorite and not some kind of from Earth origin is not so easy. Yeah. However, I've also found several round spherical structures under my stereo microscope, which are collected using magnets a few years ago. But then I talked to someone, and they said it's actually, if you really want to make sure it's a micrometeorite, you got to do some kind of an isotope analysis or whatever, and then it's getting complicated quite quickly. Freshwater biology, a user-friendly guide to the ciliates. Thank you very much. Okay. It's called Freshwater Biology by Mr. Feusner. It's a weird question, but wild plants green. I already talked about that. It has to do probably something with the solar spectrum. Okay. How about some precious still water from some places? Wonder what you can find there. Generally, what I found is in the free water you won't find a lot. You have to collect some algae, some sediment, some biofilm. Usually that's the place where you find a lot. Yeah. Chlorophyll might have evolved underwater. Okay, so yeah, so that means plants that are growing deeper in water tend to be more reddish to be able to absorb the pigment, the greenish and bluish light more. Yeah. So I recommend up to 50 microliters, but I think it's a personal preference. What micropipette capacity range? I use disposable pipettes, suck some up, drip some out while judging the amount of debris. Yeah, I've also got disposable ones are fine as well. Yeah. Here, this one I cut off simply to collect larger organisms as well. Are dinoflagellates common organisms to observe in pond water? Dinoflagellates I've observed in marine water, and a few years ago I did actually they have these nice beautiful shapes, but in pond water I've not seen them. Red is filtered out with the first 10 meters of water. The orange and yellow, okay, yeah. Then orange and yellow. Okay, that's it. From Google. Plants are green as the leaves of grass and oxygen in our atmosphere change, allowing sunlight to be filtered better with chlorophyll, which is green. Before that, most plants were purple. Okay, interesting. Well, no, the plants are able to absorb also red light, especially when the sun is low. Then the sunlight is more reddish in color, and that's also something that plants are able to absorb better. Ultrasonic cleaner tip, put the thing to be cleaned in a Ziploc bag. Oh, together with your cleaning solution, fill the ultrasonic tank with warm water, add your Ziploc bags and switch on. That is an interesting one. Then lets you use different cleaning solutions in the same time. Thank you, never thought of that. So basically what you do is you basically prevent also the cleaning solutions from mixing and maybe being diluted away, and you just put them into a Ziploc bag. That's an interesting one. Thank you. Okay, so again, about every 60 feet plus, everything is blue and green. Okay, crystals in the sand, yes. Thinking about dry sample, how would one go about finding and maybe identifying cysts? You mean cysts from, for example, from amoeba, the so-called the test data from tested amoeba. So what I would do is in a dry sample, I mean, I've got some moss over here. It's sometimes difficult to actually shake it out. But what I usually recommend is that you soak it in water and then kind of rinse it out and then collect whatever solid material you find then. Yeah. So usually, even if the material is dry, sometimes rinsing out the things are sometimes easier to do. Yeah. Scrape from the biofilm from the side of the pond, should still find something interesting. Yes. Okay. Yes, at the DIC. Vinogradsky column, not tried it yet, but is a possibility. Are the tips of some diatoms collecting radioactive elements? I mean, let's put it this way. All organisms will accumulate radioactive carbon-14, for example. Yeah, so this is actually something that happens. So I can imagine. I don't know if there's bioaccumulation happening in diatoms where they specifically concentrate radioactive elements, but I know that living things generally do that because there's this natural radioactivity around us, and there are naturally radioactive elements around us which are basically incorporated into living things. Yeah, so this does actually happen. Yeah. Perfusion chamber, no, did not try. Okay. Okay, but thank you for the recommendation. The Clostridium was in a pond. I think it was in one of the pond or river water samples. So occasionally you find them. You have to collect the algae. Yeah. Any alternatives to immersion oil? Not that I know of. I mean, I would not use vegetable oil or anything like that. Yeah. Just buy yourself a bottle of immersion oil, I would say. Yeah. I'm not saying that others don't work. I know that some companies offer microscope objectives that can be used in oil immersion as well as for water immersion. So I know that this does exist, but that would be but I would use immersion oil. Yeah. So immersion oil has a refractive index. I talked about that. Could you use image stacking? I talked about the image stacking. Okay, DIC again. Is a nice tick of activity, surely, but 60x dry suggested by Microbehunter in the video is still my goal. Seems others have done that, so yes. Yes, I think that's an important comment. I would highly recommend that, look, many microscopes, when you buy them, they come with 100 times oil immersion objective. And I think the reason is because you can then sell the microscope with a higher magnification. I really rarely use my oil immersion. Today I used it just for the sake of this live stream, but I think it's impractical to use, and you have to be really careful because once you have used the oil, put oil on the microscope slide, you cannot use the lower magnifications anymore because otherwise you get risk of getting the oil on the other objectives. So you have to be really careful, right? And especially mistakes do happen. Yeah, so I would highly recommend to actually get replace the 100 times oil with a 60 times objective. Yeah, because you're simply going to use it much more often. Yeah, and the difference in magnification is not that much. I have to also say that. Yeah, you mentioned partially sliding the filter slider to make a dark background. Okay, mine worked only partially. Yeah. Okay, so what I'm gonna do is very quickly, maybe because it's already quite late at where I am, and where do you find radiolarians? That basically you need special sand samples where you can find those. I recommend that you buy if you really want to see them, you can buy microscope slides. So there are some sand samples out there in some regions of the world that have a lot of radiolarians. Okay, for those interested in meteorites, check out the work by John Larson and his project Stardust. Thank you also for that recommendation. Okay. Yeah, and no to BSL2. Yes, I can agree. What does this mean? Yeah, Typhoon just mentioned that. No to BSL2 biosafety level 2. So this basically means that, strictly speaking, if you're messing around with unknown bacteria, you are already in biosafety level 2, which actually has some, you need to be actually a laboratory in order to do that, right? And that's also one of the reasons why I don't like doing that. And as a matter of fact, I'm a teacher. And in the syllabus guide that we're using for teaching, it actually states that we're not allowed to work with bacteria other than E. coli. Yeah, and I generally also say you got to be careful with contaminants as well, right? So, and growing human bacteria, like what you sometimes see on YouTube, people kind of comparing fingerprints and so on. I wouldn't do that, just out of safety. Yeah, yeah. So I'm just saying, okay. I think I'm just going to leave it at that. Again, it was slightly longer. It's already an hour 40. And I still have a whole bunch of samples that I did not look at. I hope that I have answered more or less all of the questions. If not, then yeah, maybe somebody else is going to answer them, or maybe I myself as well in the comments later on. I'm just going to call it a day for today. Yeah, I hope that you liked the video. Again, I'm always happy for recommendations on what to look at, so thank you very much. Also, thank you again for everyone who's donated. Yeah, all of this money goes to a separate bank account that I use exclusively for microscopy. Okay, so I try to return this somehow also to the community a little bit. Yeah, I wish you all the best. Okay, I wish you all happy microbe hunting as always, and hope to see you again next week. Bye bye.